So
the
it's
adrenal secretory, the response there in the adrenal.
yeah,
Very interesting.
adrenal and also the just sympathetic versus parasympathetic. You know how they have opposite effects on different organs. But that's nothing to do with how we think about things and what we want to know. And I don't know anything about that.
I find it very
That's
interesting.
psychology is so interesting. You don't think about the biology behind it.
Well, some of us will try to pick up the torch and keep carrying it and follow your work, and we look forward to that. I just find it interesting that Crest is such a small part to do with the visual system. The visual system's so complex and so old, and that it has its role in that system as well.
You were mentioning the ganglion cells that it makes. Is that the only part that it plays in the visual system?
Well, the ciliary ganglion innervates the eye, and it controls the size of the iris. And so there are muscles in the eye that are derived from neural crest. There's not the cornea of the eye is derived from neural crest. So there are components like that, but really the eye is CNS, it's central nervous system. So most of its components are not going to come from crest. It's just when you have the outer coverings and things like that, have contributions. But the eye is one of the simpler parts of the nervous system. That if you really want to understand CNS, I think that's exactly the place to look, because it's compared to other things, it's relatively straightforward. So.
You think the eye is straightforward?
Compared to the brain, yeah.
Right, yeah. Great. Well, that's a light at the end of the tunnel, I think. We were talking earlier before we got on with you about the bipolar neurons in the peripheral ganglion that are neural crest derived. Have you studied much their afferent and efferent functions? We're being taught that they're modified unipolar cells. So the idea of having an afferent efferent system through them is of interest to us. If you can explain.
Well, I've always thought of them as being sort of a relay. They're the first point of. I mean, they're pretty amazing because they send axons very long distances, and they sense many different things. They sense temperature. They sense pain. And they relay that back then to the central nervous system. So why need them? Compared to just having an afferent that comes out of the CNS, I don't really know. But clearly they are important because depending upon how big the structure they innervate is, the ganglion size actually changes. So, like at the limb level, the dorsal ganglia are very much larger than they are in other parts of the body. So there are interesting, sort of matching between the size of the ganglia and the size of their targets, which is great.
And I would imagine there's probably plasticity there in the evolutionary sense.
Oh, absolutely. No, there's a lot. There's a lot known about that because there are these target-derived factors that are important for survival for the neurons. So what happens is when the neurons are first born, they become post-mitotic. You have a certain number of them, but they tend to be overproduced, and then depending upon how big the target is, they either live or they die. So it's very non-conservative the way the embryo makes these ganglia. They just make these very large ganglion that then are weaned down in size depending upon what they need to innervate.
Use them or lose them.
Yeah, and it's the same thing. It's not just the DRGs, also the motor neuron pools, which are CNS, but also have afferents that go out to the periphery, are very much the same. You have very large pools in the limb-forming regions compared to the other parts of the trunk.
And is there a developmental window when that happens? Postnatal?
There's a critical period during
critical period.
Yeah, and it varies. It's different for different ganglia.
Amazing.
Yeah, that's really interesting. There was a lot of work being done on that, and in fact, one of the Nobel Prizes went for that work to Rita Levi-Montalcini, together with a few other people, because they discovered nerve growth factor and epidermal growth factor and things like that.
Right. So in a sense, these derivatives in many ways make us who we are.
I think so, yeah.
Darwinian view of it as well. Fascinating. Well, it's renewed my interest in it. I've been very interested in neural crest for a long time, and you spurred us pick up that torch because there's a lot to learn yet.
Question. Yeah, sorry. So can you use neural crest cells and stem cells interchangeably, or are they basically the same thing?
Neural crest instead of what? I didn't hear the rest of your question.
Neural crest cells and stem cells, are they interchangeable?
That's a very good question, and it's also a loaded question. so a neural crest cell in an embryo is a really not very good stem cell. So the cells are multipotent, and they can differentiate into many different derivatives. And people call them stem cells mostly because it's a lot easier to get funding if you call it a stem cell than if it's not. But in fact, by definition, a stem cell is one that can self-renew for long periods of time, whereas a real neural crest cell in an embryo goes through a few cell divisions and then it differentiates. So it's not a good stem cell. Now that said, you can take stem cells from many places and make them into neural crest cells, by having the right mix of stuff, growth factors and things like that. So, and neural crest cells can also be made into many things that you want a stem cell to be. So in some ways they can be transitioned from one to the other, but I wouldn't say that they're equivalent in any way. And in fact, in some ways I think that neural crest cells are really modified neural stem cells, and that then go on to differentiate.
So I've heard of stem cells before, but I've never really looked into them too much. I know what we can use them for, but where do you get them from?
Nowadays, it used to be that the only real stem cell was considered to be the fertilized egg, which can form all different cell types. Now, then people started making embryonic stem cells, especially from mice. They have not been successfully made from many other organisms. But in mice, you can actually grow lines of embryonic stem cells that then you can make into a mouse, for example, or into any cell type. But since then, so people started looking everywhere for where you can get a stem cell from. And the way that embryonic stem cells were made initially is they were made from germ cells, from eggs or sperm from mice. And they can be converted into these embryonic stem cells and then propagated. Now it's been found that you can take cells even from adults that can be made into stem cells. And for example, you can take skin and grow the cells for a while, and then these skin cells can be made into stem cells. And so now that's why there was all this hubbub about is it the only way you can make stem cells is from embryos, but now it's clear that you can make them from other tissue as well, and that you have these cells persisting with these stem cell-like properties well into adulthood. And from the brain, you can make neural stem cells. So there's some precursors that seem to, they sort of go dormant, but you can find them. And it makes lots of sense because muscle is a good example. Muscle is a postmitotic cell, right? So the muscles in our body differentiate into muscle, and then they actually fuse into these multinucleated cells that are very big and long. And your muscles are important, right? But if you only had the muscles you were born with, you'd be in pretty bad shape. So say you go running and you tear your muscle. It takes a while, but your muscle eventually will heal. And the way it heals is that there are stem cells in your muscle. They're tiny and they're quiescent. And only when you damage your muscle do your stem cells begin to repopulate and redifferentiate into muscle. So your body's probably filled with cells like that. And it's true in your brain also. We always tell our students that you shouldn't drink because you're going to kill off your neurons. But in fact, there is at least some ability for new neurons to form even in the brain.
Right. We've been told that it's in the hippocampus, that 6,000 you're in the dentate gyrus of that hippocampus.
Right. So that's a pretty new experiments when people have been able to show that there's new birth of these cells all the time.
We're just in awe that you're joining us here today. How many students do you have in your lab? Are they all doctoral students, master's students?
I don't have so many students right now. I mostly have postdoctoral fellows, and I have about 15 of them. I have quite a big lab. Yeah. It's expensive.
And what are you working on this month, for example?
Sorry, say again?
What are you working on this month?
Oh, mostly grants.
Right. Same old, right?
I mean, I don't do that much work myself anymore in the lab. I do a little bit, but not a lot. But it's mostly what I do is I'm helping supervise all these other guys.
Mm-hmm. And are you passing the torch to them? Are they ambitious?
Most of my postdoctoral fellows go on to get academic positions. Not everybody, but I've got a pretty good hit rate. I'm very proud of that. That's probably my greatest accomplishment is my progeny.
Thank you so much for helping us do this interview today. It's just a great.
It's my pleasure.
I have another question, just one more. What kinds of questions do you have right now, whether you've tackled them or not, that you wish could be answered, in your lab?
Oh, that's a really excellent question, and it's so big. What would I really like to know? I mean, there are a lot of things I'd like to know that don't have to do with my own research. But in terms of neural crest, I would really like to be able to figure out the whole range of interactions that make a cell undergo an epithelial to mesenchymal transition and begin to migrate. And I'd like to understand, I mean, we know something about how the early steps occur, but we know so little about what makes them migrate, how they navigate along these pathways, what makes them stop. We don't know why they end up at a particular place and then they differentiate. So if I could somehow bridge that gap between when they come out of the neural tube and then they find their site, I would really like to know that. And I think that's feasible within my scientific lifetime.
Beautiful. Looks like you've got a lot of life to live there.
No, I'm getting to the point where I'm thinking, how many more years do I have to write grants? No, I love what I do. It's really, really fun. But I have to say that the most satisfying thing for me is dealing with young people like you, and because you're the guys that are going to answer those questions. I might be, sort of saying, oh, but let's work on this problem, but you guys are the hands, and so that's how it gets done.
Thank you so much. You are awesome. Thank you very much for joining us today. And you're a real sport for joining us and helping us with launch our club.
Well, I think it's great. I love what you're doing, and best of luck.
Thank you. We'll stay in touch.
Okay, thanks.
Thanks
Take
so
care.
much. Bye.
Bye.